Welcome to part 3 for the digestive system lecture. Today we're talking about the intestines, starting with the small intestine. So the majority of the events of digestion and absorption are going to all occur in the small intestine. And the small intestine is going to extend from the pyloric sphincter in the stomach down to the ileocecal sphincter where the large intestine begins. The small intestine gets divided into 3 separate regions. We have the duodenum, which is right after that stomach. This is going to be the shortest region, and it is going to be retroperitoneal. The duodenum is going to start at the pyloric sphincter of the stomach, and it is going to be in kind of the form of a C-shaped tube that is going to extend about 25 centimeters until it merges with the jejunum. The jejunum is the middle portion of the small intestines and is about a meter long and is going to extend all the way down to the ileum. And jejunum actually means empty, and this is because when people were first starting to study the digestive system, they would look at a lot of corpses, and in the corpse, this portion of the small intestine is empty, so that's why they call it the jejunum. And then the last region in the small intestine is the ileum, and this is the final and longest region of the small intestines, and it's going to be up to about 2 meters or about just over 6 feet in length, and it is going to join the large intestines at a smooth muscle sphincter called the ileocecal sphincter. Functionally, the small intestine is going to have 3 main roles. The first is going to be the segmentations are going to help to mix the chyme, the digestive juices, with those digestive juices, and it's going to help to bring food into contact with the mucosa so that nutrients can be absorbed. And then additionally, peristalsis is also going to help to propel that chyme through the small intestines. The small intestine is also going to complete the digestion of carbohydrates, proteins, and lipids, and it's also going to begin and complete the digestion of nucleic acids. And then last but not least, the small intestine is responsible for absorbing about 90% of the nutrients and water that are going to pass through the digestive system. So lots of absorption is going to be taking place in the small intestines. Moving on then to look at the histology of the small intestines. The wall of the small intestine is going to be composed of the same 4 layers that we see throughout most of the GI tract. We've got our mucosa, our submucosa, the muscularis, and our serosa. So here we say mucosa, submucosa, muscularis, and serosa. The mucosa is going to be composed of layers of epithelium, as well as a lamina propria and the muscularis mucosae. And the epithelial layer of the small intestinal mucosa is going to consist of simple columnar epithelium that are going to contain many different types of cells. So here we look at the various types of cells that are going to make up our epithelial layer of the mucosa. First, we have our absorptive cells. These are going to contain enzymes that are responsible for digesting food, as well as they're also going to possess these little microvilli that you can see here at the top, at their apical surface, and these are going to allow for the absorption, an increase in the absorption of nutrients in the small intestinal chyme. There are also going to be goblet cells that are present in the epithelium, and these are going to secrete mucus. The small intestinal mucosa is also going to contain many of these deep crevices that are going to be lined with glandular epithelium. And these cells that line these crevices are going to form the intestinal glands, and they are also going to secrete intestinal juices. The Paneth cells are going to secrete lysozyme. This is a bactericidal enzyme, so it's going to kind of clear out or destroy any sort of bacteria that might be in the small intestines, or unwanted bacteria, I guess I should say. These Paneth cells are also going to be capable of phagocytosis, and they may have a role in regulating the microbial population in the small intestine. So this microbial population that we see in the small intestine is going to actually be very important in the process of digestion. This microbiome that's living in the small intestine is going to play an important role, which we'll discuss later. There are 3 types then of enteroendocrine cells that are going to be found in the intestinal glands of the small intestines. We have our S cells, and these are going to secrete the hormones secretin, which we'll talk about that in a little bit. We have the CCK cells, and these are going to secrete cholecystokinin, and the K cells, and these are going to secrete glucose-dependent insulinotropic peptide, or GIP. And we'll talk about what these things do a little bit more as we go. The lamina propria of the small intestine of that small intestinal mucosa is going to contain areolar connective tissue, as well as an abundance of mucosa-associated lymphoid tissue, that MALT that we talked about before. And solidary lymphatic nodules are going to be the most numerous in the distal part of the ileum. So that's where we're going to find most of these lymphatic vessels here. And then groups of lymphatic nodules are going to be referred to as aggregates of lymphatic follicles, or Peyer's patches, and they are also going to be present in the ileum. The muscularis mucosa of the small intestine, of the small intestinal mucosa, is also going to consist of smooth muscle. Moving forward then with this histology, the submucosa of the duodenum is going to contain duodenal glands. These are also called Brunner's glands, and these are going to secrete an alkaline mucus that's going to help to neutralize that very acidic gastric acid that's in the chyme. So it helps to sort of neutralize the pH there, so that absorption can take place more readily, and so we don't damage those cells in the intestine. So within the small intestine, there are going to be these circular projections, or these called circular folds, also called plicae circularis, and these are going to be permanent ridges that are in the mucosa, and these are going to help to enhance absorption, and they do this by increasing the surface area, and causing the chyme to sort of flow in a sort of spiral fashion as it passes through those small intestines, and this is going to allow for increased absorption to occur. So here we can see our circular folds here. And you can see it, this is a little bit of a more expanded image here. The mucosa of the small intestine is going to form these finger-like villi. And these are going to be these deep folds that you can see. They're sort of finger-like, next to these circular folds here. And these are going to, finger-like folds. These are going to, again, help to increase the surface area of the epithelium that's available for absorption and digestion. So much of the histology that's associated with the small intestine is really going to be related to some sort of functionally going to be related to the process of absorption and digestion, specifically absorption. So embedded in the villus is going to be a lacteal. This is a lymphatic capillary that you can see. It's animated here in green. And this is going to be important for fat absorption. The free surface of the absorptive cells is going to also feature microvilli, and these again are going to increase the surface area, and they're going to form that brush border that we've seen in the microscopes many times before. Before the microvilli, because those microvilli are going to hugely increase the surface area of the plasma membrane, the larger amount of digested nutrients is going to be able to diffuse into absorptive cells in a given period. So this increase in surface area is really going to be beneficial when it comes to increased rates of absorption, or increased amounts of digested nutrients absorbed. The brush border is also going to contain several brush border enzymes that are going to have an assortment of digestive functions. So looking at those intestinal juices and those brush border enzymes, intestinal juices are going to provide a vehicle for the absorption of substances from the chyme, and they're also going to come into contact with the villi. Some intestinal enzymes, like those brush border enzymes, are going to break down foods inside the epithelial cells of the mucosa on the surface of their microvilli. And then some of this digestion is also going to occur in the lumen of the small intestine. So looking at the mechanism for digestion in the small intestine, we've got segmentation. This is going to play a really important role in digestion. So segmentations are localized mixing contractions that are going to occur in portions of the intestine that are distended by a large volume of chyme. Segmentations are going to mix the chyme with the digestive juices, and they're also going to bring the particles of food into contact with the mucosa so that it can be absorbed. And then they are also going to, so they can be absorbed. Sorry. However, it's important to mention that these segmentations are not going to push those intestinal contents along our digestive tract. That's going to be done by propulsion. All right, so it's important to remember the difference between peristalsis, which is a propulsive kind of contractions, and segmentation. All right, this is for mixing and digesting. Peristalsis is for moving things along, for propulsive contractions. Right? So it's important to understand and remember the differences there. So, looking at the differences between these peristalsis and segmentation. Segmentation is going to be localized. These are mixing contractions that are going to occur in portions of the intestine, like we said, that are distended by these large volumes of chyme. Segmentation is going to mix the chyme with the digestive juices, and it brings those particles of food into contact with the mucosa for absorption. Segmentation is not going to move intestinal contents along the tract. Peristalsis is going to be the contractions that are going to propel or move those intestinal contents along the GI tract. Migrating motility complexes is going to begin in the lower portion of the stomach, and this is going to be what's responsible for pushing chyme forward along that little short stretch of small intestine before it dies out. So that the MMC, or migrating motility complex, is going to slowly migrate down the small intestine. It's going to reach the end of the ileum in about anywhere from an hour and a half to 2 hours. And then at this point, then another MMC is going to begin in the stomach. So this whole peristalsis process of peristalsis is going to be fairly slow. That brings us then to the digestion of carbohydrates chemically, how they're broken down in the small intestine. So carbohydrates are going to be broken down into monosaccharides for absorption, and these starches are those starches that are not already broken down into their disaccharides, like maltose, malteriose, and alpha dextrins that are going to be broken down by the salivary amylase. So there's any starches that are left over that aren't broken down into disaccharides are going to be cleaved by those pancreatic amylase that we mentioned last class. The brush border enzymes are then going to break down the disaccharides into monosaccharides. All right, alpha dextrin is going to be broken down into glucose by alpha dextrinase. Maltose is going to be broken into glucose by maltase. Sucrose to glucose and fructose by sucrase. And then lactose is broken down into glucose and galactose by lactase. So, chemical digestion of proteins then. The protein digestion is going to start in the stomach, where proteins are going to be fragmented into peptides by the action of pepsin. All right. The enzymes in the pancreatic juices, the trypsin, the chymotrypsin, the carboxypeptidase, and elastase, are going to continue to break down those proteins into further broken down into peptides. And then the protein digestion is going to be completed by two peptidases that are in the brush border of the small intestines, called aminopeptidase and dipeptidase. As far as the digestion of lipids is concerned, this is going to be or the breaking down of fats, most lipid digestion in adults is going to occur in the small intestines, and this occurs using an enzyme called lipase, or a group of enzymes called lipases. Bile salts, which are amphipathic molecules, are also going to break the globules of triglycerides, those little flat droplets, into little droplets, and this is a process called emulsification. The pancreatic lipase, due to the increased exposed surface area of those droplets, can then hydrolyze more triglycerides into fatty acids and monoglycerides. So here we have just a basic table summarizing the digestive activities of the pancreas, the liver, the gallbladder, and the small intestine. And we really talked about the pancreas, the liver, and the gallbladder in the last lecture. So this is a nice summative sort of slide. We also have a cute little table looking at a summary of the digestive enzymes and the source that's producing them, the substrates that they're breaking down, and the products that they result in. And then we also have clearly more digestive enzyme tables. Okay, so now moving on to look at the absorption that's going to occur in the small intestine and how this happens. So now that we've talked about the digestion of the various types of nutrients in the small intestine, let's look at how each of these different types of nutrients are absorbed. This is a really great little image that kind of depicts nicely all of the different things and the different ways that the various nutrients are absorbed. So we can see here that our simple sugars, our monosaccharides, are going to use facilitated diffusion or are going to use a secondary active transport with sodium. All right, they're going to move into the cell, and then they have facilitated diffusion into the blood capillaries of the villus. This is going to drain into the hepatic portal vein. This is eventually going to end up in the liver. Fructose is, so we've got our galactose and our glucose using secondary active transport with sodium, and then fructose is able to use facilitated diffusion. With our amino acids, we've got a couple of different options. With simple amino acids, these are going to be able to utilize active transport or secondary active transport with sodium. And then our dipeptides and our tripeptides are going to use a secondary active transport using hydrogen ions. Once these amino acids have entered the cell, they are going to be able to diffuse out of the cell again into the blood capillaries of the villus. This is going to flow empty into the hepatic portal vein, which is going to lead to the liver. Next, we have our small short-chain fatty acids and our triglycerides. Our small short-chain fatty acids are able to use just simple diffusion. They're going to slide right on through that cell membrane, and then they're going to diffuse out the other side, where they will, as we've said before, empty into the blood capillaries of the villus. That's going to flow into the hepatic portal vein. That's going to empty into the liver. Now, looking at the micelles here, this is going to do something a little bit different. Right? So this is how we're going to allow for the triglycerides, the large short-chain and long-chain fatty acids, and the monoglycerides to enter into the cell. So these are going to form a micelle, which we can see here, and they're going to make use of that sort of amphipathic properties. They're going to have a simple diffusion across into that cell, where they're going to then, it's going to lead to the disruption of that micelle, and then this is going to lead to the formation of a chylomicron. We'll talk about this a little bit more in a second. This is going to leave the basolateral surface, and this is going to be transported to the lacteal of the villus, and this is then going to move to the thoracic duct, which then is going to lead to the junction of the left and internal jugular and left subclavian veins. All right? And we'll talk about this whole process in a little bit. So, looking at the absorption of lipids and bile salts. Dietary lipids are going to be absorbed by simple diffusion. We've discussed that, nice and simple. However, these long-chain fatty acids and our monoglycerides are going to be absorbed as part of a micelle. They're going to be resynthesized into triglycerides and are then going to form this protein-coated spherical mass called a chylomicron. These are going to be taken up by the lacteal of the villus, and then from that lacteal they're going to enter the lymphatic system and are then going to pass into the cardiovascular system, where they're going to reach the liver or the adipose tissue. This is an interesting little process here. So, looking at the absorption of alcohols, because this is a really interesting process. This is one of those reasons that it's not a good idea to ever drink on an empty stomach, and it's because of this whole process that we'll discuss right now. So, the intoxicating and incapacitating effects of alcohol that you may or may not be familiar with are going to depend on your blood alcohol level. And because alcohol is a lipid-soluble solution, the absorption of alcohol is going to begin in the stomach. However, the surface area that's available for absorption is going to be much, much greater in the small intestines than it is in the stomach. So when alcohol is able to pass into the duodenum, it's going to be absorbed much more rapidly than it would be in the stomach. Therefore, the longer the alcohol remains in the stomach, the more slowly blood alcohol levels are going to be able to rise. So because fatty acids in chyme are going to slow gastric emptying, blood alcohol levels are going to rise more slowly when fat-rich foods, things like pizza and burgers and nachos and fries and all that other good yummy stuff, are consumed with your alcoholic beverage. So there is a scientific reason why you should have pizza with your beer or whatever you're drinking. Also, the enzyme alcohol dehydrogenase is present in the gastric mucosa cells, and this is going to be responsible for breaking down some of that alcohol into acetaldehyde. This is not going to be intoxicating, although it will give you a doozy of a hangover if you have too much of it. So that's going to be, this alcohol dehydrogenase is going to be very important for breaking down alcohol so you don't just get totally drunk. When the rates of gastric emptying are slower, proportionally more alcohol is able to be absorbed and converted to acetaldehyde in the stomach, and therefore less alcohol is actually going to reach the bloodstream. Given identical consumptions of alcohol, females are often going to develop higher blood alcohol levels and are therefore going to experience a greater level of intoxication than you would see in a male of comparable size. So if we have two people, male and female, both of the same weight, same height, etc., female will often experience greater intoxication, have higher blood alcohol levels than the comparable male. And this is because the activity of gastric alcohol dehydrogenase can be up to 60% lower in females than in males. Now, obviously, this is not always the case, and there are also going to be important things to consider with this alcohol dehydrogenase level, specifically regions of the country, of the world. Some races are going to have genetically are just going to have lower levels of alcohol dehydrogenase than other countries. It's an evolutionary trait that is actually quite interesting. Specifically, in some Asian populations, even Asian men will code for equally low levels of alcohol dehydrogenase. And this is an interesting kind of correlation because in these same populations, there was an increased risk for GI type cancers or esophageal cancers, and this is also associated with increased drinking, or alcohol consumption. So if this lack of alcohol dehydrogenase basically meant that this group of people were getting drunk faster, but it was also reducing the levels of alcohol that they were consuming, which helped to reduce their risk of esophageal cancer due to alcohol consumption. Kind of interesting. So there's some interesting correlations there. So here we've got just a nice little pictogram showing the movement of nutrients from the GI tract in the small intestine, basically terminating and ending in the liver. We can see the absorption of our fatty acids, the various amino acids, our simple sugars, being absorbed into the venules here, and then the absorption of the chylomicrons containing our fatty acids into our lacteals and how they move through the body. So, I'm not going to spend too much time on that. So moving forward then, looking at the anatomy of the large intestine, also called the colon. The large intestine is going to extend from the ileocecal sphincter all the way down to the anus. It's going to be subdivided into the cecum, the colon, the rectum, and the anal canal. Hanging inferior to the cecum is going to be our appendix, and as you can see right here, our appendix. And then the colon is going to be divided into a ascending colon, transverse colon, a descending colon, and the sigmoid portion, which we see here. So, moving forward. Functions of the colon. This is where haustral churning is going to occur. We'll talk about that in a minute. As well as peristalsis, and mass peristalsis is going to drive the contents of the colon into the rectum. The bacteria that are found in the colon are also going to convert proteins into amino acids. They're going to help to break down those amino acids, and they're also going to produce some vitamin B, or B vitamins. Then this is also where absorption of water, ions, and vitamins is going to occur, as well as the formation of feces, and then the ultimate act of defecation. So just a silly little joke. What happens when you eat aluminum foil? You sheet metal. Just kind of silly dad jokes. So anyhow, looking at the histology of the colon, the wall of the large intestine is going to have the same typical 4 layers that we find in the rest of the GI tract. We've got our mucosa, our submucosa, our muscularis, and our serosa. The mucosa is going to consist of simple columnar epithelium, as well as a lamina propria, again made up of areolar connective tissue, as well as, the muscularis mucosae, or which is made up of smooth muscle. Looking at the histology of the colon more closely, looking at the actual cells that make up this the colon, the tissue. The epithelium that's in the colon is going to contain mostly absorptive cells and goblet cells. The absorptive cells are going to function primarily in water absorption, and the goblet cells are going to secrete mucus that's going to help to lubricate the passage of the colonic contents. So both absorptive and goblet cells are going to be located in these long, straight, tubular intestinal glands called crypts of Lieberkranz, crypts of Lieberkranz, also called tubular intestinal glands. These intestinal crypts, we can call them. These are going to extend the full thickness of the mucosa. The salivary lymphatic nodules are also going to be found in the lamina propria of the mucosa, and these can extend through the muscularis mucosae into the submucosa. The mucosa of the large intestine is going to have no villi and no perpendicular folds, and no permanent circular folds. It does not have a simple columnar epithelium with, it does, however, have a simple columnar epithelium that's going to have numerous goblet cells. The muscularis is going to contain specialized portions of the longitudinal muscles called the tinea coli, and these are going to contract, and they're going to gather the colon into a series of pouches that are called haustra. So polyps of the colon are going to be slow-growing, benign growths, and these are often going to be removed, as they can sometimes become cancerous. So this is why it's so important to get your colonoscopy after you get to a certain age, to check for these polyps. So looking at the mechanisms, or the mechanical digestion that's going to take place in the colon. So one movement characteristic that we see in the large intestine is called haustral churning. And this is in this whole process, the haustra are going to remain relaxed, and are going to become distended as they fill up. When the distension reaches a certain point, the walls are going to contract and squeeze the contents into the next haustra. Peristalsis is also going to occur in the colon. This is going to be, however, at a slower rate than in the more proximal portions of the tract. So this is going to be anywhere from 3 to 12 contractions per minute, so it is slower. The final type of movement is mass peristalsis that we see in the colon. And this is a strong peristaltic wave that begins at about the middle of the transverse colon and is going to quickly drive the contents of the colon into the rectum. So because food in the stomach is going to initiate this gastrocolic reflex in the colon, mass peristalsis is usually going to take place about 3 to 4 times a day, or immediately during or after a meal. So moving then to the chemical digestion that occurs in the colon. The final stage of digestion is going to occur in the colon through the activities of bacteria that are going to inhabit the lumen. So this is a really important gut bacteria. The mucus is going to be secreted by glands in the large intestine, but no enzymes are going to be secreted. Chyme is going to be prepared for elimination by the action of bacteria, which are going to ferment any remaining carbohydrates and release hydrogen, carbon dioxide, and methane gas. The bacteria are also going to convert any remaining proteins in amino acids, and they're also going to break down the amino acids into simpler substances like indoles, skatols, hydrogen sulfide, as well as fatty acids. Bacteria are also going to decompose the bilirubin into smaller pigments called stercobilin, and this is going to give feces its kind of characteristic brown color. The bacteria that are in the colon are also going to produce products that are going to be absorbed in the colon, and these are going to include several different types of vitamins that are needed for normal metabolism. These are going to be things like B vitamins and vitamin K. So, moving on then to the absorption and feces formation. For absorption, the colon is going to absorb water, electrolytes, and then some vitamins. And feces is going to consist of water, inorganic salts, as well as some sloughed off epithelial cells, some of those gut bacteria, the products of those bacterial decomposition, and any undigested portions of food. So although most water absorption is going to occur in the small intestine, the large intestine is going to absorb enough water in order to make it an important organ in maintaining the body's water balance. So patients who have had large chunks of their large intestine or their colon removed for one reason or another are going to need to be particularly watchful for any signs of dehydration. Even though most water absorption does occur in the small intestines, enough of it does occur in the large intestines to where this is going to be an important role for the large intestine there. So, last but not least, we have our defecation reflex. This is going to occur in a series of steps that we're going to discuss right here. So, in response to distension of the rectal walls, the receptors are going to send sensory nerve impulses to the sacral spinal cord. The motor impulses from the cord are going to travel along the parasympathetic nerves back to the descending colon, the sigmoid colon, the rectum, and the anus. These resulting contractions of the longitudinal rectal muscles are going to shorten the rectum and thereby increasing the pressure within it. So this pressure, along with the voluntary contractions of the diaphragm, as well as the abdominal muscles, plus some of the parasympathetic stimulation, are going to open the internal anal sphincter, allowing for defecation to occur. So, we've talked about all the various parts of the GI tract. So let's look at the different phases of digestion. All right. First, we have the cephalic phase. This is going to stimulate gastric secretions and motility. Next is the gastric phase. This is going to be a neural and hormonal mechanisms that are going to be involved. And then we have the intestinal phase, which is also going to involve neural and hormonal mechanisms. So, the cephalic phase. This is during this phase of digestion, the smell, the sight, the thought of, the initial taste of food, all of those things that go along with eating, are going to activate the neural centers of the cerebral cortex, the hypothalamus, and the brainstem. The brainstem is then going to activate the facial, glossopharyngeal, and the vagus nerves. And the facial and glossopharyngeal nerves are going to stimulate the salivary glands to secrete saliva. So this is why your mouth kind of waters when you smell something really good. And then the vagus nerve is going to stimulate the gastric glands to secrete gastric juices. The purpose of the cephalic phase of digestion is just basically going to be to prepare the mouth and the stomach for the food that's about to be eaten. Next, we have the gastric phase. This is where neuronal regulation is going to begin, and this happens when the stomach walls are distended or when the pH increases because proteins have entered the stomach and are buffered. Because proteins have entered the stomach and buffered some of the stomach acid. The stretch receptors and the chemoreceptors are activated, and this is going to result in waves of peristalsis and the continual flow of gastric juices. The hormonal feedback for this is going to be a negative feedback loop. It's also going to regulate the gastric secretions during this gastric phase. Chemoreceptors and stretch receptors are going to stimulate the autonomic nervous system to release acetylcholine, and this is going to stimulate the release of gastrin by those G cells. Gastrin is going to stimulate growth of the gastric glands as well as secretion of large amounts of gastric juices. It's also going to strengthen the contractions of the lower esophageal sphincter, and this is as well as increasing motility of the stomach and relaxing that pyloric and ileocecal sphincter. Our final phase of digestion is the intestinal phase. This is going to begin when partially digested food is going to enter the small intestine. There are two components to the regulation of this. We've got neural and hormonal, just like with the gastric phase. So in the neural regulation of the intestinal phase, this is going to be stimulated by distension of the duodenum, and this distension is going to trigger the enterogastric reflex, which is going to reduce gastric emptying. The hormonal regulation of the intestinal phase is as follows. The intestinal phase of digestion is going to be mediated by two major hormones that are secreted by the small intestines. All right, we've got our cholecystokinin and our secretin. Secretin is going to promote secretion of bicarbonate ions into pancreatic juice and bile. It inhibits the secretion of gastric juices as well as promoting normal growth and maintenance of the pancreas. It's also going to enhance the effects of cholecystokinin. So overall, secretin is going to cause a buffering of acids in the chyme. Our cholecystokinin is going to stimulate secretion of pancreatic juices that are rich in digestive enzymes, and it's also going to stimulate the ejection of bile into the duodenum. And this is also going to slow gastric emptying. So that is all we have for the digestive system. Here are the links for the Wiley Plus. And as always, if you have any questions, comments, or concerns, please feel free to email me or see me after class.